Good evening, everyone. This is Jesse Silvera with Spirit of the Coast Analytics. All participants are currently muted, and this presentation is being recorded. I do not plan on having a question and answer at the end of this call. However, you can email me any last questions you may have, and if there is enough overlap, I'd consider making a second video sometime over the next 2 weeks to address them. Many of the topics and themes that we'll go over in this video will be made public sometime over the next 2 weeks with more in-depth analysis. To begin, we are going to start with a very quick 50-second promotional video by Anavex. This video was first presented at the 2025 annual shareholder meeting, but I know its distribution has been pretty limited. We'll then follow the promotional video with a recording of Dr. Sabbagh's presentation today. I know this has been circulating. Some of you may have watched it in entirety by now, but it should give us a good baseline for the last segment of my presentation, which is in PowerPoint form. So without further ado, Anavex's annual shareholder video.
Imagine a world where memories don't fade away. There's a new potential oral treatment called blarcamazine that could change the game for Alzheimer's. It's not just another pill, it's a lifeline. This treatment fits seamlessly into daily life, making it easier and safer for patients. It's like pressing pause on time, giving families more precious moments together. No more constant worry or feeling helpless. This might be a giant leap towards a future where Alzheimer's doesn't dictate lives. It's not just about managing symptoms, it's about preserving the essence of who we are. Ready for a world where memories last longer? The future is here, and it's brighter than ever.
And now we have Dr. Sabbah's AAIC 2025 presentation that he gave this morning.
I'm going to today present the phase 3 attention AD study, long-term data over 3 years of continuous treatment with blarcamazine, oral blarcamazine to significantly show you benefit in early Alzheimer's disease patients. In this capacity, I chair their scientific advisory board of Anavex. These are my disclosures. Blarcamazine, I hope you're starting to get come around to the idea of a sigma-1 receptor agonist and autophagy. So this is a new mechanism of action. This is not an amyloid-dominated mechanism or a tau-dominated mechanism, but this is the sigma-1 receptor agonist. What we're seeing is a reconceptualization of the concept that we thought amyloid was the earliest seminal event that triggered everything downstream, but there's now some suggestion that impaired autophagy, which leads to lysosomal and synaptic dysfunction, could be a triggering event for the accumulation of amyloid and the downstream pathology. So this is kind of a reconsideration of how the chain of events occur. When we look at the fact that impaired autophagy can have adverse effects, maybe it's an early event. We understand that it could be driven by environmental factors, genetic factors, but it could be preceding the accumulation of plaques and tangles or other pathology. And as a consequence, you have reduced plasticity and other changes as well. So the conceptualization here is that restoring autophagy through sigma-1 receptor activation could restore normal function and neuronal function. So I've shown this slide on a few presentations before, but the conceptualization is that the accumulation of amyloid and tau and the vesicle formations and maturation and lead to impaired lysosomal function, or which of course is lysosomal dysfunction, and the consequences you get aggregation of the proteins, which consequently leads to neurotoxicity, and that if you gave an oral sigma-1 receptor activation, you would restore autophagy by leading to normalization of lysosomal function, which then causes the docking, autodegradation, and neuroprotection and homeostasis. So today I'm going to show you some of the long-term data on the basis of their phase 2b/3. We published this data, the paper, January of this year on their phase 2b/3 of Anavex's blarcamesine program. And the take-home messages here were that it was oral, once daily, novel mechanism of action, and a good safety profile, which I will emphasize later. So today we're going to talk about how we got to the long-term data, and this is the long-term beneficial therapeutic effect. So the original study that was published in the McFarlane paper in JPAD of January of this year was 2 doses, 50 milligrams a day, 30 milligrams a day, or placebo, oral medication once daily, about 170 per group, and that was the randomization for 48 weeks, and then you rolled into the open label extension. The primary outcome measure of the original study was ADCS, ADL, and ADAS-Cog, with a key secondary outcome of the CDR sum of the boxes. We also did a pre-specified analysis on some of the gene variants, including the gene variant for the sigma-1 receptor. In the original paper, in the original study, this is this side of the paper. This is the blinded phase, placebo. This was in the ITT. This was 340 or so. This is 170 or so, and then we rolled them into the open label extension. And so the placebo became open label, and blarcamesine had a continuation. We followed these people for up to 3 to 4 years. The important critical elements are long-term safety was demonstrated at 4 years, 192 weeks. There was no new emergent events. The TEAEs, the treatment emergent adverse events, tended to be mild, nausea and things like that. No ARIA events, no deaths related to it, no adverse toxicity on vital signs or liver toxicity, and most of the TEAEs, being dizziness and things like that, were transient. And when you adjusted the dose down, it went away and you could continue the treatment. So the adherence was excellent over a long period of time. We understand effectively we've created a delayed start analysis, and kind of unintentionally, one of the delayed starts analysis was COVID-19. And so some of the patients were in the randomized blinded phase and then had a delay in getting to the open label extension. So a part of this group was continuous treatment in the open label extensions. Part of it had effectively a washout for a while. So we actually had some variability in the restart in the open label extension. So I'm going to show you the population of people who were continuously treated and then the population that had an interruption. So this is the, I'm going to show you, so on the left-hand side, this is the randomized phase. This is ADAS-13, pre-specified ITT, but I want to be very clear that in the pre-specified ITT, 2 points delta on the ADAS-13 was seen in both 50 milligrams and 30 milligrams. You did not actually need to combine the cohorts to get a therapeutic efficacy benefit. This hashed line is actually the participants in that washout phase because of COVID, but then the reemergence of the treatment effect, and after 4 years, the delta has gotten bigger and bigger at 3.83 points. When you actually look at the same group but no interruption, so this is the group of people that did not have any effective washout but continued, that effect actually was 4.2 points on the ADAS after 4 years, a significant, highly statistically significant in both groups, but even more significant. So this is making this long-term argument that continuous treatment had continued sustained long-term benefit. So ADAS was one of the 2 pre-specified endpoints, which looked at the ADCS-ADL. So in the original report, when we designed this study, remember at the time this study was written, you needed to have 2 co-primary endpoints. And so we had an ADL activity outcome, and we had a cognitive outcome with ADAS-Cog13. So the reason I say this to you is that in the McFarlane paper, we reported that the ADAS-13 hit on this pre-specified endpoint, the ITT analysis, but the ADCS-ADL did not. Numerically favorable, but not statistically significant. When you follow these patients out, this is that treatment interruption hatched line. You still saw after 4 years a very, very significant clinical benefit in ADCS-ADL, and that benefit was much more magnified in patients with no treatment interruption. So this light blue line is patients who got blarcamesine continuously in the blinded phase and the open label extension phase, and you see that they had a significant benefit over time. So the point being is that we see a sustained benefit, and what you didn't see at the blinded phase, you ended up seeing later on in the open label extension. When we look at, so this is effectively a delayed start analysis. I don't think it was intentionally designed this way, but just what happened with COVID is that you see a great safety profile, no new treatment safety toxicity events. The titration was manageable. Dizziness, nausea, you just downward titrate the dose, and you could get an effect. They would go away. No deaths related to study drug. You saw a continuous, sustained, robust effect that continued over 4 years, did not wash out. And this was sustained over time with a p-value on ADAS-13 of 0.0165 and ADCS-ADL of 0.02606. It's suggesting potentially a disease-modifying effect, and also supporting the idea that early initiation had a sustained effect and continued long-term benefit over time. So we actually also have now gene variants, and I said that in my earlier slide. We have the gene variants, and what we know is that if you have this wild type of the sigma-1 receptor gene variant, which is 70% of all of us, actually the effect is even bigger. So 30% have a gene variant, but 70% are wild type. And when and this was pre-specified, is that the gene variant, the wild type variant, actually had a more robust effect over time. So we can now use precision medicine approach to determine who's going to get an even bigger approach, which is also in and of itself a very novel finding. And you see that when you do a time analysis or time saved analysis, you see that you can save almost 2 years. In conclusion, blarcamesine is a once daily oral medication that restores autophagy through sigma-1 receptor activation. When we do a pre-specified gene analysis, we understand the wild type has a better effect, but we also see that the clinical benefit is quite sustained, and it can work over time. And when we look at the long-term benefit, we see good safety, good toxicity, clinical meaningful treatment effects, no ARIA events, long-term benefits on up to 4 years now on ADAS-Cog 13. And interestingly enough, now we start to see clinically statistically significant benefits on ADCS-ADL. I want to thank all of my co-investigators. I want to thank our patients and our participants and the study staff. This is not a study that gets done unless hundreds of people worldwide are participating in this, and we're grateful for everyone. Thank you.
All right. So I hope you enjoyed listening to Dr. Sabbagh. I particularly enjoyed his exclamation to the crowd early on about coming around to sigma-1 and autophagy as a therapeutic, as well as the way he described lysosomal dysfunction as preceding amyloid burden, which is what I believe to be true, and his enthusiasm regarding the robustness of the data. So this is a slideshow I just put together going over AAIC 2025 observations, which has a heavy emphasis on Anavex. As a reminder, I'll be creating an AAIC special topic paper on my website over the next couple of weeks to wrap up these themes in greater detail. So we're going to cover the sigma-1 wild type in the OLE. We're going to cover the time saved analysis, and what's next, before concluding with just a few kind of rapid thoughts on other aspects, compounds, and companies that I viewed at AAIC. As a disclosure, I am long AVXL. I'm not a financial analyst, and you should do independent due diligence when making investment choices. So this slide gives a quick recap on what we know coming into the week. We knew that by the completion of the primary 48-week study, cognitive results when assessing ITT patients in placebo arm were statistically significant and clinically meaningful, and uh, we see that here. After a washout period, when placebo arms swapped to intervention, the open label extended to 192 weeks, which is a bit over 3.5 years, comparing the full-time intervention group to the former placebo group in what is called early start and late start. In this analysis, we see that blarcamesine extended cognitive benefit, especially in early start patients, and we see that with the delta of negative 4.20. This aligns with the drug's mechanism of action, lysosomal dysfunction, especially through age-related alkalization and pathological gene mutations. It's at the beginning of disease pathogenesis, and the drug works best by battling this dysfunction on a flat field. So if the disease is left unattended for too long, the drug now fights an uphill battle with substantial lysosomal bloating, autophagy cargo buildup, and pathological burden through extracellular and intracellular amyloid and tau. Falling over to the right side of the slide to ADCS-ADL and function, there was a positive trending at 48 weeks, but it was not statistically significant, and divergence between early and late start increased tremendously by the end of the OLE at 192 weeks, showcasing a statistically significant and likely clinically meaningful result, though clinically meaningful thresholds for function are less defined. Personally, I think it makes sense that changes in your brain morphology, the cooling of pathological burden, and returning the brain to homeostasis would result in a slower functional improvement. In addition, I got to witness a pretty stellar presentation earlier on in the conference on lysosomal dysfunction, which found that it took an extended period of time for autophagy enhancers to make an impact in preclinical models. It's probable that in a more complex model, i.e. humans, the cascade from intervention to be made meaningfully, meaningful autophagy changes to improving lysosomal volume, function, and target, to improving pathological load and downstream function. It probably takes a significantly longer period of time in humans. So when Anavex presented their Sigma-1 wild-type OLE slides, they did not have a visual representation. This gives opportunity for confusion since there are multiple efficacy slopes that could be produced to create the numerical benefit given. I spoke with Marwan and Kunjin after the presentation and confirmed that the early start group, which is the light blue line on these charts, the light blue line, relatively plateaued. So these two large charts that you're seeing here are ADAS-Cog13 and ADCS-ADL respectively, and I personally altered the slope from week 144 to week 192 in each one of these to show what this plateauing may look like. And these are totally notional since it's likely that the entire OLE slope actually looks different. So not just from week 144 to 192, which is what I changed, but the entirety from week 48 to 192 probably looks different than this. What I do want to point out, though, is that early administration resulted in a solid curve and plateau, indicating an incredible disease modification. Here, I just show you these notional graphs compared a bit larger to their genuine ITT counterparts. So on the left side, we have ADAS-Cog. In the top left, we have the notional chart that I created for you that kind of showcases what a plateauing may look like against its genuine ITT in the bottom left. And then on the right, we have ADCS-ADL. Again, in the top right, we have the notional chart that I created showing a plateauing effect. And then on the bottom right, we have the genuine ITT slides. And I'll just sit here for about 10 to 15 seconds for you to look at this really quick and kind of make some comparisons before moving along. Okay, so here I present the ADAS-Cog and ADCS-ADL metrics numerically. I show that for cognition, all early patients jumped 107.2% in efficacy between 48 weeks and 192 weeks. Sigma-1 wild-type only patients extended that benefit an additional 29.3%. For function, all early patients jumped 641.9% between 48 weeks and 192 weeks, which is primarily due to a lag effect, the lag effect in the efficacy on function. And Sigma-1 wild-type only patients extended that benefit an additional 65.2%. Earlier and sustained access to care halts cognitive and functional loss over time, especially in 72% of the global population, though all patients benefit regardless of their genotype. Here we go over time saved analysis. Anavex states that 3.68 years of treatment, right? We did the math on how many weeks that is. 3.68 years of treatment confers 1.62 years of time saved to patient. So this means that the patient maintained cognition and function in earlier, less intensive disease stages. So it's saving these patients from slipping into moderate or late stages of the disease. Meanwhile, 1.5 years on donanemab, also known as Kisunla, conferred approximately 5 months of time saved. And 4 years of lecanemab, or Leqembi, confers about 1 year of time saved. So looking at this chart here, we see that Anavex provided 44% 44% time saved when measuring time on drug and time benefit, the access between how long a patient had to be on the drug to receive that time saved benefit. Meanwhile, donanemab only provided 27.8% and lecanemab only provided 25%. Doing some quick math, we can see that Anavex's time saved potency is 58.2% better than donanemab and 76% better than lecanemab. There's some key takeaways. Obviously, Anavex's blarcamesine distinguishes itself by being widely efficacious regardless of genomic makeup. There's no constraints that we are aware of by gender, APOE type, or comorbidity. The drug is safe, orally available, easily transportable. There's no need for monitoring, and will likely be less expensive than existing medications, probably between 10 and 20,000 USD annually. In Sigma-1 wild-type patients, earlier treatment confers comparatively 76% more time saved than lecanemab and 58.27% more time saved than donanemab. This analysis is important because it validates the pre-specification of Sigma-1 as an efficacy booster, which informs insurers and payers on why they should pay for blarcamesine. So if a patient has Sigma-1 wild type and is in early stages of the disease, an insurer should have no issue justifying the coverage of blarcamesine. Furthermore, it gives patients options. So if I'm a patient and I have the Sigma-1 mutation, perhaps I opt out of treatment knowing that I maybe won't perform as well as someone that had the wild type, even though, all patients benefit. Maybe I'd rather go with a separate therapy altogether. And so you can inform payers, insurers, and patients with this precision medicine effect. And that's really the criticality and the importance of the Sigma-1 pre-specification in the OLE and the data that we received today. So what's next? We know through conversations with Marwan, Nell, and Hank that there is a new GWAS paper. We know that they discovered something powerful, and we know it's right around the corner, possibly third quarter around August. It's highly likely that the additional gene discovery is related to Col24 alpha 1 based on a redacted ADPD abstract last year. Col24 alpha 1 is a collagen family gene, and personally, I think it's a little unclear from the abstract, which is still available. I maintain it in my records. How many patients have the Col24 alpha 1 wild type gene? But I read it as about 50% of patients. There may be some debate on that, but I read it as about 50%. So patients that had the wild type basically doubled in efficacy compared to those that didn't. And you can see that from the ADAS-Cog and CDR sum of boxes scores that I present on the screen. ITT, all patients benefited about -2 points against placebo for cognition. But when only accounting for Col24 alpha 1 wild type patients, this benefit extended to a whopping 3.9, -3.9 points on ADAS-Cog, so almost doubled in efficacy. Similarly, CDR sum of boxes improved for all patients by about -0.48 points, and Col24 alpha 1 wild type patients improved to about -0.99 points. So again, almost about doubling in efficacy. So one would theorize that patients with Col24 alpha 1 wild type and Sigmar1 wild type had explosive results in the study representing a super responder kind of situation. Both Nell, Marwan, and Hank were very excited for this data to publish, as you can see from some of the quotes on the slides. Strong signal, very exciting. And right before I left session today, Hank caught me in the hallway and said, just wait until you see it. We should also be expecting the OLE data to be compared against AD&I very shortly. Wolfgang mentioned that he had carefully completed the analysis last week, and it was simply pending Kunjin's statistical review, basically. Comparing OLE against AD&I is standard practice, and it was reassuring when he stated that his findings, Wolfgang's findings, were both striking and robust. As far as the EMA process, Marwan broke character and showed visible enthusiasm when he said that it was going great, and it was confirmed that Audrey's post-monitoring poster was requested by the EMA. Notably, it was relayed that Dr. Missling recently met with the FDA director, Marty McCreary. Notably, further information, no further information was provided on that, but the company is in initial stages at the FDA. They were all aware of the new commissioner's voucher, and when I asked if they would at least attempt to apply for it, they all reaffirmed that they were aware and interested, and their body language told me that they're probably going to try for it. If they don't get it, we probably won't hear about that. So there's no guarantees there. Furthermore, Anavex's new U.S. Medical Affairs staffer, David, mentioned that he knows Marty personally and has his cell phone number, and they plan on engaging or leveraging some of these personal relationships. Finally, on the regulation front, it was an interesting tidbit. Marwan dropped when he mentioned that regulators in the Middle East were looking at blarcamesine. We know he briefed blarcamesine in the Middle East recently, and it's not really clear how far along the regulators in the Middle East are with blarcamesine. It's not clear if they've submitted to the Middle East or if they're just in kind of introductory stages there. Personally, I would not expect any new trials soon. We may get some. I wouldn't expect any. It was clearly relayed that Alzheimer's is the top focus of the company, and that is where their limited resources are going. I suggested that a partnership would speed up the progression into other indications. There was some kind of uncomfortability there based on this, as well as a comment Nell made about possibly being able to move on to new projects next year. It's my opinion that the company will have partnered with BP or been bought out shortly after EMA decision. I cannot guarantee this, and it's totally speculation on my part, but there's yeah, some emerging or converging points that make me feel this. Separately, I requested quality of life updates on the Alzheimer's program. I'm not sure that we will get this because it may be considered too promotional by the EMA, which is something that the EMA is kind of hard on. But Wolfgang stated that it's notable in that long-time patients that have been on blarcamesine for years and years, still handwrite Dr. Missling letters requesting access to the drug, and that the drug is making meaningful improvements in their lives, and that it's notable that they can even still write at all, considering their age and their affliction. There are a couple considerations to the data presented today. I think most notably is that by week 192, there is very possibly some survivor bias. meaning that the lower sample sizes of patients being assessed at week 192 are, most likely the patients that responded best to drug. They have maybe the best overall health or had the least side effects on the drug. The flip side to this, of course, is that with a smaller sample size, it's much harder to reach statistical significance, and the tiny p-values that we receive today indicate a high magnitude of effect. To close out, I have a couple very brief slides on other developments at AAIC. I will go more into detail with these with my formal paper since I need to do a little bit more research on them. But first up is cognition therapeutics, which I know the most about. They gave a really great recap on their dementia with Lewy bodies, their Shimmer trial. It was a phase 2 trial that showed a really incredible breadth of effect across the entire disease from motor skills to cognition, neuropsychiatric, and daily living. And the company is expecting a decision about a breakthrough designation from the FDA by or before September this year. Additionally, there was substantial interest in TMEM106 Bravo mutations as it relates to both longevity and lysosomal health, as well as a TREM2 agonist being developed by Muna Therapeutics. Um, TREM2 agonists have kind of had a bit of a mixed bag, some mixed results, but um, mechanistically and delivery-wise, their therapy looks like one of the most promising in its class. Muna is currently a private company. And finally, I got a good presentation on Violet Therapeutics' VT001, which is an EphB3 modulator. Basically goes after inflammation, and this could feasibly be paired with basically any therapy to tackle inflammation. So you would kind of pair this on with almost any drug that you wanted, whether that's blarcamesine, whether that's any of the monoclonal antibodies or anything that emerges in between then and now. Um, and finally is Alnilem. I may be mispronouncing that. Alnilem Pharmaceuticals. It's the only public company mentioned in this presentation besides Anavex and Cognition, which is progressing some state-of-the-art RNAi technology to target APP. The preclinical work was really very convincing, with excellent APP knockdown and long-term amyloid reduction. They will need to verify these biomarker improvements actually confer some sort of cognitive or functional benefit, but this is something that I will be at least keeping on my radar for a while. So I really want to thank everybody for their, you know, contributions to sending me out here, all of my benefactors. It means a lot that you guys trust me to come out here and get information for you. I hope that you found any of the information in this presentation useful and in the donor chat that we had set up. Again, if there are any questions that you have, please send them to my email. I'll try to get to them. If there are enough questions and there's enough overlap, I will make a second video and send that out to the group. So I believe that is it, and I hope everybody has a really good night. And thank you very much. Appreciate it.